Unlocking nature's secrets. Can mushrooms and other fungi help beekeepers in maintaining the sustainability and survivability of honeybee colonies? Over the past decade, there has been a significant decrease in the populations of both native pollinators and honeybees. Most recently in 2025, it was reported that commercial beekeepers lost more than 60% of their colonies. This equates to 1.7 million colonies lost at an estimated cost of more than $600 million in the United States agriculture. Honeybees contribute up to $15 billion globally. Obviously, this has a tremendous impact on agriculture worldwide, as more than 1/3 of our commercial fruits, nuts, and vegetable production, more than 130 different products, are dependent on the honeybee for pollination. But why the recent steep increases in pollinator and honeybee losses? Previous studies looked at large-scale impacts from pesticide exposure and habitat loss. Although these factors play a role, scientists agree that this pressure is stable and was not the cause for the increased winter losses of honeybee colonies. In fact, it has been determined that beekeeper colony management plays the most important role, especially in regards to pest management and nutritional support, both which can be supported by the beekeeper. Because of the massive winter colony die-off, in February of 2025, the USDA scientists collected bee and colony samples from California and other western states. These samples were sent to and analyzed at the Bee Research Laboratory in Beltsville, Maryland. The focus of the research was to try to find what parasites, pathogens, and viral notifications were in both the living, dying, and dead bees. What the researchers found was that in the dead and dying colonies, there were high levels of deformed wing viruses, both A and B types. There were also high levels of acute bee paralysis virus. These viruses were found in both individual sick bees and pooled colony dead samples. Deformed wing virus affects all stages of honeybees. The severity of the virus depends on when the individual bee is infected. If the bee is infected as an adult, i.e., an infected mite feeds on that adult, then they will appear normal and are rarely affected, but they can transfer that virus to an uninfected mite, and that mite can then infect other bees. Regardless, all 3 stages of development affect the sustainability of the honeybee colony. Acute bee paralysis virus is a RNA virus that causes significant, often fatal symptoms in adult honeybees. These symptoms include trembling, inability to fly, and you'll see hairless, black, greasy-looking bee bodies. It's a leading cause of colony collapse, often spreading through crowding or fecal oil routes in the hive, with symptoms that can appear 5 days after infection. So how are these viruses getting into the hives and killing our bees? It is well known that the Varroa destructor mites are a vector for these viruses. The mites are picked up in the environment by the bees, taken back to the hives, where they then feed on adult bees, infecting the adults, or dropped into the larval cells where the larva develops and they infect the developing larva. Commercial beekeepers were treating for these mites. The problem is, when they investigated the mites and tested them, 100% of the mites were resistant to amitraz, the active ingredient in the apivar strips. And what does this mean for beekeeping? Well, it means that we need to find new and innovative ways to treat mites and the viruses they carry to make healthier bees, as well as promote bee sustainability for the future of agriculture. As a beekeeper in the Pacific Northwest, I'm fascinated by the natural beauty of my surroundings and the interactions between organisms, especially those that might be affecting my girls. As you may know, honeybees and other native pollinators are indicator species. Their populations and behaviors reflect the overall health of our ecosystem, as they are highly sensitive to environmental alterations. A frequently overlooked dynamic is the intertwined relationship the pollinators, and especially honeybees, have with indigenous fungi. In my training as a beekeeper, I was initially only informed about the parasitic effects of fungi on honeybees. The two most notable interactions were chalk brood and nosema. Chalk brood is caused by a common contagious fungi, Ascosphaera apis. It is recognized by finding hard, white, or black mummified larvae in the comb or at the bottom of the hive entrance. It is prevalent in spring, often triggered by stress, cold, high humidity, or weak colony populations. While it rarely destroys a colony, it weakens them, reduces honey production, and is managed primarily through improving colony nutrition and hygiene. The other fungal disease, nosema, is a serious disease which has two different forms affecting European honeybees. Nosema apis, this is historically the most common form in cooler climates. It is often associated with dysentery and crawling bees that cannot fly. The other form is Nosema ceranae. It's an invasive species that originally came from Asian honeybees. It is now the dominant form worldwide. It is very deadly and can cause colony collapse without showing classic signs like dysentery, regardless of which type of nosema is present. It causes digestive issues, population decline, queen loss, and potential colony collapse. Honeybees, however, proved just how much beekeepers did not know about the symbiotic relationship between honeybees and fungi. In 2018, a mycologist named Paul Stamets, a mushroom scientist, made an amazing discovery. He noticed honeybees foraging on mushroom mycelium, those thread-like networks fungi grow beneath the soil. It looked like the bees were drinking droplets from the fungi, almost like they were self-medicating. So Stamets and researchers from Washington State University tested extracts from two common forest fungi, the reishi mushroom, Ganoderma resinaceum, and the amadou, the Fomus fomentarius mushrooms. The results were astonishing. Bees fed these mushroom extracts had up to an 800-fold reduction in deformed wing virus. And in field tests, colonies that were treated with Ganoderma showed a 45,000-fold drop in another deadly virus, the Lake Sinai virus. Think about that. 45,000 times less virus just from mushroom tea for bees. Nature had created antiviral compounds long before we even knew what viruses were. Then, 5 years later, scientists in the Czech Republic discovered yet another fungal ally, the gypsy mushroom, Cortinarius caparatus. When honeybees were fed a simple alcohol extract from this mushroom, it completely prevented the development of deformed wing virus, both in lab cages and in live field colonies. Even better, the bees' immune systems became stronger. Their lifespans were unaffected, and the honey they produced was pure, no chemical residues, no side effects. This was something beekeepers had dreamed of for decades, a natural, safe, and sustainable antiviral treatment. For the first time, mushrooms weren't just decomposers, they were defenders, a line of protection for one of Earth's most important pollinators. But what about the mites themselves, these vectors of the virus, those tiny vampires that destroy the hives? At Washington State University, entomologist Dr. Steve Shepard and his team, including Jennifer Hahn and Nicholas Nager, found a way. They took a soil fungus called Metarhizium, already known to kill insects, and trained it through directed evolution to survive inside a warm hive environment. Normal fungal strains died at 95 degrees, which is the typical hive temperature. But after 2 years and 27,000 mite tests, the Washington State University team created a heat-resistant strain that could live and thrive in hives. Once inside, the fungal spores germinate, penetrate the mite's body, and kill it from the inside out, leaving the bees completely unharmed. The next steps are already underway, getting approval from the Environmental Protection Agency and then developing a practical delivery method for beekeepers. We hope that in the next decade, beekeepers can replace chemical miticides with natural fungal solutions. And the implications go far beyond bees. The same directed evolution techniques used to adapt this fungus could make other biocontrol agents stronger, helping protect crops without chemicals, restoring balance to agriculture. And remember, this is a story not just about fungus and honeybees, but it's a tale of resilience, collaboration, and the incredible intelligence of nature. It's a story about our honeybees, tiny, tireless creatures that pollinate 1 out of every 3 bites of food we eat, from almonds to apples, from coffee to cucumbers. Their work sustains our diets and our entire agricultural system. It's never been more important for beekeepers to observe their bees, be in their hives, watch their behaviors, listen to what they're trying to tell us, and smell what they're trying to communicate with us. This is especially important because your primary responsibility as a beekeeper is to be an observer, to see what the bees are doing and anticipate what they might need. The bees will let you know. Only then do you become the manager, intervening to provide what the bees need to increase their sustainability.